Hi Fatima, this is Ines. So this will be a brief overview of G-proteins and as well as apoptosis, which is cell death. So if you've got any questions or you want to practice any exam questions together, feel free to let me know. And I'm happy to cover that for next session. Otherwise, we'll start. So with a G protein, we first need to have a good grasp on what cell signaling is, and we did ascertain in our last class that there are three main stages of cell signaling. So the first one, which we can see here in this diagram, is reception, and this obviously happens when we've got a signaling molecule or a ligand which attaches itself to a specific receptor. This chemical signal is detected when the signal molecule binds to a receptor protein located on the cell's surface. And we also distinguished in our last lesson that this can occur either intrinsically or extrinsically. Extrinsically or intrinsically. So either the receptor can bind with the ligand on the cell wall or inside, and that is dependent upon whether this signaling molecule is either hydrophilic or hydrophobic. We understand as well that in the second stage, which is signal transduction, the binding of the signaling molecule changes the receptor protein in a way that will initiate transduction. And this is basically a series of further signals within the cell, and this occurs in the cytoplasm. And this basically serves as a chain of communication. Now, obviously, we don't have to know the signal transduction pathway in detail because it is fairly complex and it's what we study in at a university level, but we do have to appreciate that this occurs and it can happen in multiple steps within the cell. And again, these molecules in the pathway are often called relay pathways. The third step that we covered is our response, and this is the third step. Basically, the transducted signal finally triggers a specific response within the cell. And this response can be almost any imaginable cellular activity, whether it be catalysis of an enzyme, for example, in glycogen, or a rearrangement of a cytoskeleton, the production of a protein, activation of specific genes in the nucleus. This cell signaling process helps to ensure that the crucial activities that is needed by required cells happen in the right cells at the right time and in proper coordination within the cells of the organism. Now that we understand this, we have to then go and ask what type of mechanisms of the cell occurs. So we understand that the signaling molecule behaves as a ligand, which is the term for a molecule that specifically binds to another molecule. So basically, the ligand binding is similar to the binding of an allosteric regulator to an enzyme. So this causes the shape change, okay? And in this case of the signal transduction, the binding of the ligand alters the ability of the receptor to transmit signals, okay? So most signal receptors are on the plasma membrane, and these are proteins, okay? And their ligands are water soluble and are generally too large to pass freely through the plasma membrane, and that is why they have to bind to a receptor on the cell membrane. Now, there are certain receptors in the plasma membrane, okay, that will bind to water soluble signaling molecules. All right, so there are three major types of membrane receptors that have been identified. We've got G-protein, or the G-protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. Okay, so we don't need to necessarily explore in detail how the mechanisms work in relation to a G protein. We just have to understand that it is a type of plasma membrane protein that enables a water-soluble signaling molecule to bind to the specific receptor and then initiate signaling transductions, okay, which will then lead to a response. This is just a brief diagram on what occurs, but basically we can see here that we've got a G protein, we've got a signal, and then this initiates a series of transductions within the cell, and then this initiates a cellular response. So that's basically it. I hope that answers your question. If you don't understand anything, feel free to let me know. Okay, so next we're just going to briefly cover apoptosis, which is cell death. Okay, so cells have to interact in such an elaborate way, but what happens when they're damaged or infected? Obviously, if we have a couple of cells within our body that is damaged, we have to get rid of it. So there is a programmed cell death or cell suicide within our body that is called apoptosis. I'm not too sure if you know about seppuku, which is a ritual suicide performed by a samurai, and it is performed in accordance to the Bishodu, which is a samurai code of conduct. In that same way, cells have a programmed suicide where they kind of die or sacrifice themselves for the greater good of the whole body. Okay. Cells also have a lifespan as well. So once they reach the end of their lifespan, they will often enter apoptosis as well. Now here we can see a diagram of a white blood cell. Okay, and this white blood cell is undergoing apoptosis or cell death, and this is what we call blebbing. Okay, so I would just want you to note the comparison between the normal white blood cell, which is on the left, and a cell undergoing, or the same white blood cell undergoing cell death. And these little bulbs or balls are called blebs. Okay, and they eventually shed as membrane-bound cell fragments and are then recycled by the body. Okay. Okay, so in the same way that in a spiritual sacrifice done, just as I touched before, it was believed to have been done for the greater good of the community, so protecting one's honor. We have this intrinsically within our bodies. So apoptosis protects neighboring cells from damage that they would otherwise cause. If that dying cell, for example, that dying cell can leak its damaged contents in the surrounding environment. Things like digestives and other enzymes which will cause further damage to the surrounding cells. Another example is when the body recognizes infected cells, there is a type of the body undergoes apoptosis, which then kills off these infected cells. And I'm not too sure if you know the show Cells at Work, but in episode 3 it covers this in details. But yeah, it's a very interesting episode. So just generally during the process of apoptosis, we have cellular agents, okay, which chop up the DNA and fragment the organelles and other cytoplasmic components. The cell shrinks and becomes lobed. And we could, as we saw in the white blood cell, the same process is happening in cancer cells as well. Okay. So obviously we don't need to go into detail as to the specific mechanisms that is initiated and during apoptosis, but I guess we just need to appreciate that it is complex and that there is a series of steps that occur. And just like we discussed in cell communication, there is a certain signal which is sent to that damaged or infected cell, and that certain signal will then initiate the apoptotic signaling pathway within that cell, and this will then initiate cell death. Okay. There are certain cancer cells which evade the immune system, and they don't undergo cell death. And sometimes these cancer cells evade the immune system, and if this occurs, this will then grow into a tumor. We do not know why cancer cells can evade the immune system, but I hope this covers and shows us an appreciation of how important apoptosis is. So it's not all bad. And again, apoptosis, it is something that is organized. The cell contents are packaged into smaller pockets of membrane for garbage collection by the immune cells. And again, apoptosis removes cells during development, eliminates potential cancerous and virus-infected cells, and it helps to maintain balance in the body or homeostasis. Okay, so apoptosis is a very good thing that happens in the body. And that's basically all you need to know of this process. And I've got a brief overview of apoptosis, but basically to show that there are 3 stages of apoptosis. So we've got the initiation stage, and again, initiation is when there is a binding of a signaling molecule, and this signaling molecule can either be intrinsic or extrinsic. Okay, so with the extrinsic apoptotic pathway, it's when we have a signaling molecule and it binds onto a death receptor onto the cell, and again this will a signaling transduction will occur within the cell, and this will then activate a series of pathways, goes to the mitochondria, and the cell will then die. Again, we have intrinsic apoptotic pathways, and it occurs. However, there isn't a death receptor on the cell membrane. And then we've got the 2 stages, which is execution and phagocytosis. So execution is the death of the cell. Phagocytosis is when we have phagocytes, which can be considered cells which belong to the immune system, and they then take away these blebs or garbage bags for collection. And again, here's just another diagram to show the stages of apoptosis. In this picture, we've got a comparison of a mouse embryo. So obviously we know that when babies are born, they do not look anything like the initial stages of an embryo. And that is because as the embryo develops, one of an essential occurrence is apoptosis. So we can see the development of the mouse paw, and we note that it is a bit webbed. Okay, the same thing happens as well in human embryos. So obviously, when a human embryo or a mouse embryo is born, they do not have webbed hands. So the reason why they are not born with webbed hands or feet is because in where the stars are located, the process of apoptosis occurs. And apoptosis is only initiated again if there are certain signaling molecules that are received by the cells here. So everything happens in an orderly fashion. Now, sometimes if this doesn't occur, I'm sorry for a big picture of a foot on your screen, people can be born with webbed feet, webbed toes or fingers. And this is because when they were developing within the embryo stage, they did not, for some reason, the cells that were here didn't receive the signals for those cells to die, and hence why the person has webbed toes. Now we covered briefly the stages of apoptosis. There is also something called a cell cycle. Okay, so basically every single cell has a certain timing or a lifespan. Okay, so the timing and rate of cell division or the growth of certain cells in different parts of the plant or an animal, these are crucial to normal growth and development and maintenance. And there are different frequencies of cell division for different types of cells. So for example, the frequency of cell division within human skin cells divide more frequently throughout their life, whereas liver cells maintain the ability to divide but keep it reserved until appropriate when necessary. Okay. So here we've got the different stages